welcome to day 99 of day 0 to breakthrough. Instead of dwelling on last week's disappointing result, the team are determined to keep moving forward. Why? They have 2 good reasons. First, the next steps in the process are vital to the vaccine development pipeline and need to be tested under pressure, regardless of whether or not the vaccine is effective. Second, the team maintain a cautious optimism that further assay optimization will lead them to a positive result for Q or Z. Today, I'm joining the team at the National Biologics Facility, or NBF, to see what their role is in the pressure test. I asked Martina, the operations manager, what it is the NBF does.
So the National Biologics Facility here at AIBN assists researchers in trying to get their biotherapeutic candidates or vaccine candidates to clinical trial. So we'll help with producing small amounts of protein that they can use for proof of principle studies, then also help the researchers get to a process for manufacturing for clinical trial.
I joined Courtney, a member of the upstream processes team, in the back lab, the super sterile room that Varsha did her transfections in way back on day 29. I had to suit up in all the appropriate PPE and subject my phone and mic to a healthy dose of ethanol before I could get through the airlock. What the team has done so far is perform stable transfections of Q and Z into mammalian cells. This is different from the transfections that Varsha performed, as the process is a bit more controlled and will lead to cells that produce our vaccine candidate in a predictable way. The team is currently in the process of scaling up their cell cultures, which means they're incrementally increasing the size of the vessel the cells grow in, working towards producing a 50-litre batch. While the cells expand, the team must monitor them carefully and check them regularly for things like viability, which is what Courtney is doing now.
Viability is a measurement of how many live, healthy cells exist within a population of cells.
She's currently preparing a small batch of cells to undergo a production study, which will give the team an idea of how much vaccine antigen their cells are going to produce. This will help them prepare for growing and purifying the vaccine in bulk. Before I left, I asked Courtney about her perspective on the pressure test and what it means for the team at the NBF.
No matter what happens, we'll be able to demonstrate that we can start with the DNA and end up with a 50 litres of a product, and then do our downstream development, show that we can purify that product out and do our analytics to show this is the product that it's supposed to be, all of that in any situation.
I asked Courtney if she'd experienced any challenges during the pressure test and if the team had learned anything during this process so far.
It's like there's always setbacks, and so as long as we're aware of where those will be. Like for instance in this one, their viability wasn't as good on the day that I checked them, and they took a couple of extra days to recover. And that adds a few days, but as long as you're aware that that's a possibility, you can build that into your timeline, and that's what we've kind of been trying to do.
Next time we check in, we're going to be joining Diego and Connor as they continue with another important aspect of the vaccine development process: antibody discovery. See you next time as we continue our journey from day 0 to breakthrough.